Good morning, everyone. All right, so today I want to talk about a few things. As the number of students who are getting quarantined starts to increase a little bit again, we're going to start doing things with like we did last year. And I think I kind of mentioned this already. But what I'm going to try to do is between my biology classes throughout the course of the day, I'm going to try to get at least one recording. Well, I'm only going to take one recording, okay, of the class. Now what it is going to be like me standing in front of the screen, pointing at things, or in front of the board, pointing at things. I'm not saying I'm the next Preston Plays on YouTube, okay? I'm not going to get as many views. I'm not going to get a lot of like and subscribes, and that's okay. I'm okay with that, okay? But what the intent of this is in the event that you aren't able to join us in a particular class period, you can at least see and hear what we did. Again, quality, maybe you're not going to be the best quality, but you should be able to see it. You should be able to hear it. We'll try to take some pictures of stuff we have written on the board, post those on the bottom as well, and I'll show you where all that is going to be. So today is Friday, September 10th. So Friday, if we scroll through Canvas, we forgot to publish. I forgot to. We have the agenda for Friday, September 10th. Now, I know a lot of you haven't been clicking on that agenda. We've just kind of say, hey, we're going to be working on this assignment, and underneath that agenda is the assignment indented. That's by design. But within the agenda for each day, tells us what we're going to do. It has links directly to a PDF if it's like something we're going to work on in class. It will have a link to the assignment, which would then have a PDF or Google Doc that is something that you can work on outside of class. So if you say, I absolutely hate paper copies, this is how you can get to it. If you say, I absolutely hate marking up a PDF and you're at home, maybe you have access to a printer. And you say, I don't. I'm sorry, then you're marking up a PDF, okay? That's what it's gonna be, okay? If you don't have access to printing, we've showed you how to mark up a PDF, we've showed you how to mark up a Google Doc, and we've given you those options, and we will give you those options. So this wonderful recording that I'm doing right now is gonna get linked in today's agenda, and it will be linked right here. Here is a link to today's in-person agenda. If you were not able to attend, please watch the video below. If it's not there, it will get posted soon. Now that's been on there every day so far this semester. Today is the day, make sure I'm actually recording. Yes, I am. Today is the day that I will put the first link up there, okay? Again, not gonna be amazing, but it's gonna be better than just missing a whole day. When I reference some of the images from the board, on the board we have a lot of pictures that we're gonna go through today. We'll try our best to remember to take a picture, and we'll just post those pictures right below it, and it'll also be under today's agenda. So some key things that we're writing down, some notes that we're going to take, you know, maybe we're filling out a sheet in class together. We'll take a picture and we'll put it on there too. Just so we're kind of aware where to access some of this stuff in the event that you are not here. Now, that doesn't mean you got COVID and you're gone for 2 weeks. It might be I have a dentist appointment and I only missed your class. Same idea, okay? Questions on any of that? Yeah.
So, like, if you're supposed to be, like, signing in for WebEx, like, since you, like, if you have COVID or something like that, or you're...
Okay, I think I know where...
Stuff, is there a class that you can log into so you can actually view the class live? My
plan is not to do a live WebEx, okay? Because trying to coordinate which class you're in and which, if you request it, we can try to figure it out. But my hope is by doing this, maybe we can avoid the WebEx just from the trying to coordinate. Because that could mean I have to potentially have 3 different WebExes going throughout the course of the biology, which is good because then you can ask questions. However, it makes it a little bit more technologically, it makes it a little bit harder. So my hope is that doing this video is going to eliminate that WebEx. Doesn't mean you can't contact me when you're gone. Doesn't mean we can't talk. Doesn't mean we can't WebEx for 10, 20 minutes to answer any questions that you have about stuff. We can just coordinate that. Okay?
Cross that bridge when we get there.
Yeah.
If we do.
Yes. Okay, so now let's talk about yesterday. Yesterday, we went through a presentation, and it was in the agenda yesterday, and it is, I'll show you where it is. It's under Thursday's agenda, and it was this one. We will use this presentation in class, and that presentation in class talked about the different types of, or what a good research question is and what a good hypothesis is. We went through that, we went through some examples, and when we set you off to work, we had put this slide and says, in your lab, you're going to try to figure out if Aspermater influences the germination rate of radishes. You need to write a research question. You need to write a good hypothesis. However, when we got to the lab, we struggled. We didn't recall and that's partially on us. Like, hey, this is what a good research question is. This is what a good hypothesis is. Just mimic that. Where we didn't maybe remind you that, hey, you have access to this, and if you have a question about what it is, here it is. After your class, second period class, we changed the template a little bit. And what I have here now on that, if you were to go make a new copy, I'm not saying you should go make a new copy of your lab report second period, but this is the format we want your research questions in. It's also on the board over there, okay? If then for a hypothesis, will the blank affect the blank, okay? Independent variable, dependent variables. We need to make sure that a research question is a question, okay? We need to make sure a research question is in fact a question. Yesterday, there were some research questions that were statements. It needs to be a question to be a research question. It's not a yes, no. Some people said yes. You gotta ask the question. So will the independent variable, whatever your independent variable is, plug that in. It's a fill in the blank. Influence or effect, you use the term that you prefer. The blank, you insert your dependent variable. Your hypothesis is an if-then statement with a prediction. So if, what's your independent variable, then what's your dependent variable, will make a prediction. Is this a prediction? Today, it might be sunny or it might not be sunny. Is that a prediction of what today's weather is going to be?
No, because you're not stating exactly what you think. You're just saying, oh, this could happen or this could happen.
Right, you're listing the options. That's not a prediction. So in your radish seed, it might or might not influence germination. Is that a prediction? No, that's a list of what might happen. Okay? Now, 2 days ago, we introduced independent, dependent variables. Don't drop, don't drop, don't drop, don't drop. We introduced independent, dependent variables. I'm pretty sure those are not new terms for you. I'm pretty sure throughout your school years, you've heard them. They're on the board. In your lab report, there's a data table that we're asking you to fill out that has you simply list these things. Well, I don't have experimental group. These are the things we're asking you to list. I don't know if I thought of this yesterday. I don't know if it's the first time it's been thought of. I don't remember thinking about it this way. But for the independent variable, maybe we did this last year, I just don't remember. For independent variable, I keep saying, what are you going to change? But forget that. What did I change in the experiment? It's the independent, what am I changing in my experiment? Okay, what is the thing that I'm changing in my experiment? The dependent variable is what we are measuring or observing. Our controlled variable is what do we keep constant? And there should be lots of controlled variables or standardized variables. Control group, I forgot to write experimental, but the control group is a group that gets no treatment at all, and we use it as a comparison. Okay, so if I'm gonna say, does fertilize impact the growth of tomatoes, I should grow a tomato plant with no fertilizer to see if fertilizer makes a difference at all. Because if my one tomato plant grows this tall with no fertilizer, and the one with fertilizer grows this tall, did it have an impact? Well, maybe not, right? However, if I only say, hey, look, this is my tomato plant with fertilizer, but I don't have anything to compare it to, how do I know whether it made any difference, right? So that's what a control group is. It gives us something for a point of comparison. The experimental group, I didn't write it on the board, is the one that does get that treatment. So it would be the one that does get the fertilizer in my made-up example.
So in that case, wouldn't you not want to try to measure the height, because that's not really what you want. You want the actual product from the tree.
But
how that impacts.
That's just a made-up example. Okay, so it's not a perfect experimental design. It's just a made-up one. Maybe we want to look at the size of the tomato, the height of tomatoes, whatever the case is. We need a control group. We need something to compare it to. Okay, so today there's a couple things we're going to work on. One of the things we're going to work on is your lab report. Okay, your lab report that you have a copy of is ready to be completed through your procedure. What's a good title in a lab report? I'll tell you what a bad title for your radish seed lab report is. Radish seed lab. That's a bad title. And you say, oops, I typed that. That's fine. Delete it. We'll fix it. Okay? A good title in a scientific paper is it tells you what you're going to read about. Because if you can say radish lab or radish seed lab, I'm going to learn about radish seeds. Okay, it's not a description. So, like if we were going to go through and we did the fishing lures the other day as an example, and we're going to write a paper about it. We're going to write a lab report. A good title would be, does the color of a fishing lure have an impact or affect the amount of muskies caught on Lake Wausau? It's a descriptive title. A person can read that and say, hey, I want to read this paper because I want to know if color impacts muskies on Lake Wausau. It's not like colors of fishing lures, because I hate muskies. They're the silliest fish to catch in the world. Panfish are where it's at. So I don't want to read that if I'm a panfisher person, because I don't really care about muskie. Okay, the title should be descriptive. So make sure your title is descriptive. Independent and dependent variables need to be in your research question and your hypothesis. Make sure you follow the format that we have listed on the board. Your materials, we modified this a little bit after you as well. This is the format we want for second period. We changed it by a fourth period. I want a list of everything in your bag, and I want each bag listed separately. What do you have in bag 1? What do you have in bag 2? What do you have in bag 3? Maybe you don't have 4 bags. Then delete bag 4, right? There is no such thing as bag 4 in your thing. But use that. Don't say water. How much water? Don't say I put some seeds. How many of what kind of seeds? If I'm going to do your research, if I'm going to mimic your research, I need to gather all the materials ahead of time, and I need to know exactly how many seeds you had. I need to know exactly how much water you had. I need to know exactly how much paper towel you had. Everything that's in your bag needs to be itemized. Okay? Itemized means every item is listed independently. Okay? The thing that you're going to go through and identify these, we're saying a minimum of 3 variables. That's what I was referencing. The last thing I want to talk to you today about in this lab report is your procedure. A procedure is like directions in a recipe. It tells you what to do. Every single thing to do. Very detailed. description of what to do. Okay? I put some seeds in a bag. That's not descriptive. I need to be able to grab your lab report, read your procedure. Oh, I take one piece of white paper towel. Okay. I placed 82 seeds in it. Okay. Radish seeds. 82 radish seeds in it. I folded it up so the radish seeds stayed in. I put X amount of water in. I put X amount of aspirin in. I put blah, blah, blah. I put it in, folded it, I sealed it, I put it in a drawer. I need to be able to do what you did by reading what you said.
Your procedural write-up should allow someone with no experience having seen or worked on your lab to recreate it just as you did.
So if we were to hand it to, say, Mr. Engelbretson across the hall, hey, Mr. Engelbretson, you don't know what this lab is about at all. He needs to be able to do the lab based upon what you tell him to do. That's how detailed it needs to be.
So just keep that in mind as you're writing it. Like you're giving someone instructions. Hey, this is how you would do what I did. That's the mindset to have when you do your procedure right up.
And then that's where we're at this point. Okay? And in Canvas, I'll show you really quick. It's in today's agenda. Design and set up your, oh, this is yesterday's, sorry. Yesterday's agenda. Due by Sunday, so we're saying by the end of the weekend, we want your radish seed lab report completed, right here, S1, radish seed lab report through your procedure. We want you to have everything done on your lab report through that part. It's going to be blank underneath. That's okay. We're not done with our lab yet. But our first standard is setting up a good, paraphrasing, good experiment. And that's what you've done so far. So that's what we're going to evaluate you on thus far is can you design and set up a good experiment? And that's through the procedural steps. This is a submission that because it's a Google Doc, follow the process to submit a Google Doc. Okay, I understand that this will change later. I thought I had a link. I will try to go back and link on how to, a screenshot of how do you submit a Google Doc again in case you forgot. But that will get submitted via through Docs, share and export, send a copy to Student Canvas, Biology, S1 lab report through procedures, submit. Okay, we're saying we want that done by Sunday at 11:59 p.m. The end of the day ends up. When you come to class on Monday, that should be done. On Monday, we'll collect data. Okay, and then that's S2. That's the analyzing, interpreting that data that we then collect. Okay, questions about your lab report, specific or general questions that I can answer for the class. Otherwise, I can address individual questions in about 2 minutes.
Let's say that we were trying to see if the way we folded up the paper towel, we would see it.
I'm going to interrupt you.
If
That's not what our experiment is, though. Okay? So if you want to, we can discuss that privately.
Yeah.
But that's not what the class set up as our experiment.
Yeah.
Okay? So, other questions about your lab report? Procedure, you guys understand what you have to have turned in. Okay, then on the way in, some of you may have, some of you may not have, and that's okay. We will get to that we'll give you some time. There's a couple things for you to work on. Today, procedure on your lab report. Have it done through the procedural stuff. If you haven't taken your quiz and gotten 100% on your safety quiz, you should take your safety quiz again. There's also this, and it's been linked a couple different days. Today is the day I finally published it because we haven't gotten to it. Puzzle Lab, Mr. DeBoer. We'll talk Puzzle Lab here quick. Some of these activities, they're referred to as a POGO. It's a company publishes. You've probably done some of them in the past. There is a paper copy on the cart. There is a PDF copy linked on Canvas. The linked PDF has 5 pages. It says right here, only do questions 1 through 11, which is on pages 1 through 4. If you're going to do the PDF. We only gave you the 4 paper pages. A POGO activity is meant to be a pretty self-contained activity that has needs very little prior knowledge. The questions being asked are coming directly from a graph or a figure. The questions being asked are coming from a paragraph that says, like, read this. You should probably read that. Read the introductions, okay? It introduces vocabulary. This one happens to be about experimental variables, reinforcing your independent, dependent, controlled variables, standardized variables, okay? So there's 11 questions that we're going to ask you to work through. Your answer should be complete. We aren't saying they have to be in complete sentences, but they should be complete, not necessarily not just single word responses. Okay? Mr. DeMore and I will talk about the due date of said POGO, and we will make that final announcement once we address that. Okay? So that's something you're going to want to grab today. Make sure, we'll talk about due date, whether it's Sunday or Monday night. I'm leaning a little bit more towards Monday night at this point. Let's just say, we're going to say it's Monday. We will give you some class time to work on it as well on Monday, okay? But it'd be something to get a start on.
Some class time.
Right, because we also have to collect data on Monday. We need to talk about graphing data on Monday. Okay, so we'll give you some time. Okay, don't expect to do it all on Monday, but we'll give you some time. So we will say it's due Monday evening is when we're going to say that's due. Puzzle Lab, I keep forgetting to talk about Puzzle Lab, so we need to talk about Puzzle Lab. The Puzzle Lab will get turned in. We did that last week, this week, Tuesday. I think we did it on Tuesday, we did it Friday. We did it last week, Friday. So 1 week ago, we did it. There are a series of questions that will get submitted in Canvas. That's your submit a picture in Canvas. Your completed front and back. There's a series of questions. We had through 4 done. Okay, I want to talk really quick about the puzzle. I'm not going to talk about the questions on the Puzzle Lab, but I want to talk about why we did the puzzle. Science is like a puzzle. That's the title. When you had 5 pieces, 5 random pieces, and you were asked to make a prediction about what the picture of your puzzle was, did you have a really good idea of what that puzzle was going to look like based upon 5 pieces and 5 pieces only?
No.
Some people maybe did. Depending upon what pieces you had in what puzzle, you may have a better idea than others. Some people didn't know what it was 20 pieces in. Okay? It really depends. Depending upon what data you get at what times might help you draw more accurate conclusions earlier in your study. You're putting your puzzle together. The more pieces you put together, right, there's a method that you have, strategy for putting a puzzle together. The more pieces you put together, how did your confidence increase of what your image was? I would hope, right? I would hope. Did you guys have every single puzzle piece?
No.
Man, someone must have lost some along the way. I don't know what happened. And I know that really messed with some of you because a lot of people's strategy of putting together a puzzle is do the edges first, and then once the edges are done, then you fill in the middle. Oops, you missed an entire side. Oops, you didn't have any corners. I wonder how that happened. The idea of that is, in science, if it was a 100-piece puzzle, if there were 100 pieces of data that would give you the perfect full image of a puzzle, of your research, do we always have all 100 pieces?
No, it's not always what happens.
Right? We don't always have every single piece of data in a scientific experiment. But what's true is the more pieces we have, the more data we have, the better conclusions, the more accurate we can, the more confidence we can have in our predictions or our conclusions. Okay? Even if you don't have every single piece. That's why we're calling it what science is like a puzzle. We were so kind as to give you a puzzle piece for a corner that maybe didn't quite match the picture. But we made a very conscious effort to say, hey, look, that puzzle piece does fit the shape. And that's to kind of represent misleading, conflicting data. Does that happen in research? Does every single data point you get have the exact same conclusion? No, right? Not always. So that was meant to be like, hey, sometimes we get some pieces of a puzzle that kind of fit, but they don't match completely. They fit, they don't match our picture. And that's what science is. But even though those 4 puzzle pieces didn't quite fit, we could still get a pretty good idea of what our picture was. Because we had 70 other pieces that said, hey, this is chickens, baby chickens by an egg carton. This is a dog by a fire truck, I think it was, or on a flag, or a chimpanzee, a tree trunk. Okay? We had enough data, we had enough images, we had enough pieces that even though we had some conflicting data, we could still draw a fairly accurate conclusion based upon the preponderance of evidence. Lots of evidence. Okay, that's why we did that activity.
Analogy makes sense? Puzzle and science?
Okay. I needed to talk about that and I forgot to keep talking about it. Our time is getting tight, but we do have time. Okay? We still have 20 minutes. We have 20 minutes of class, 18, I think, technically. In the next 18 minutes of class, I would, because due Sunday night is the lab report, through the procedure, each one of you individually is turning in a lab report. You are turning in your lab report. Doesn't mean you can't talk to your group. Doesn't mean you can't talk to your neighbors. But it's your lab report with your words, and it's your product. Make sense? That's what I'd say number 1 priority is. Safety quiz, if you haven't gotten 100% on the safety quiz, that might be a good number 2. Number 3 would be get starting on that experimental variable, POGIL, we call it POGIL. They don't actually say POGIL on them. Maybe down in the print it might say like copyright POGIL or something. Okay? And that'd be like the 3rd thing. If you have individual questions, we are definitely here to help. Let us know. If you want us kind of spread out, you are more than welcome to. But otherwise, that's what we're working on.